Using this protocol, any lab that has a standard confocal microscope equipped with a 405 nanometer laser can perform hair cell progenitor laser ablations and monitor their regeneration.
Unlike electroablation, this technique limits damage to surrounding cells and is more accessible than a powerful, pulsed UV laser setup. Confocal imaging can also be performed immediately before and after ablation. This technique enables us to better understand the regenerative behavior of sensory progenitors, which may help in the development of therapies for human hearing loss.
For mounting, first pipette 3 to 4 anesthetized larvae into a small droplet of E3 tricaine solution into the center of a 35 mm dish with a 14 mm number 1.5 cover slip bottom. Remove the excess solution so that the larvae remain in a small droplet just large enough to contain them, and place the dish on the stage of a binocular stereo microscope. Manipulate the zoom and focus so that all of the larvae are in the field of view, and use a transfer pipette to add a thin layer of agarose solution onto the cover slip. Remove the excess agarose until the liquid just fills the well at the bottom of the dish, taking care not to aspirate any larvae, and use a hair knife to quickly orient the larvae in the agarose solution with the rostral side facing left. Gently press the larvae down against the glass such that the larvae lie in profile with their right sides facing down. After about 60 seconds, the agarose will start to solidify.